You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that had, has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoy the podcast, please listen, subscribe, like, and share it with friends. Thank you. Hello, this is Richard Jacobs with the Future Tech and Future Tech Health podcasts, and I have Trevor Morin and Bill Dunbar. They're co-founders of Pore Guys. The website is twoporeguys.com. And poor not meaning no money, but poor meaning like a pores of skin in your face, for instance. So it's P-O-R-E. So TWOPOREGUIYS.com. So Trevor and Bill, thanks for coming.
Thank you. You're welcome.
Yeah, so what's the premise of Pore Guys?
The premise is we build technology products that leverage this thing called nanopores, and that's the P-O-R-E reference, nanopores, that do single molecule sensing. And we leverage single molecule sensing for a variety of diagnostic applications in agriculture, human, could be animal, could be a lot of different use cases. So it's really a platform method of doing sensing and analysis.
So is it, since your whole platform is about single molecule sensing, I don't know, but I would guess that's a very difficult thing to do up until now?
That's accurate. I think people, particularly in the research world, have wanted to examine how molecules behave at the single molecule level for a long time. And there's some fairly complex instrumentation out there to do that in a variety of ways. And the nanopore is an interesting kind of riff on that, because it doesn't tell you a whole lot about the molecule in its sort of native form, but it's very cheap and it's very fast. So it's just like is it like a tiny opening, a tiny gateway or a pore that allows only one molecule to go through at a time? That's exactly right. Yeah. And as you said, fast, and it's an electrical signal. So you're only implicitly saying that you saw a molecule. It's not like you had a photograph or an image, but you do get this signal. And really the strength of the company from an IP point of view is the things that we do to a sample, so that when the target or targets that you're interested in go through that cheap, fast sensor, you get enough information out of that signal to make inferences for diagnostics.
Maybe I'll just add.
Is the pore taking just one molecule and that's it, and it closes, or does it gate it so that it takes in one at a time and sends it through some kind of flow analysis?
Yeah, it's always open. So it's more like sand through the hourglass a little bit, although it's not flowing continuously. Molecules randomly are captured by a voltage and pulled through that pore, which is otherwise always open and always conducting current. And it's only when that single molecule goes through that you'll see an attenuation in the current transiently.
But what about does this work for polar and nonpolar molecules? And, what if you have 3 different kinds of molecules or 100 different kinds in a medium? How do you know you've got the right one?
Yeah, that's the art, right? So we play with the buffer, we can tune pH, we can tune temperature, we can tune voltage, we know the charge of the surface of the nanopore itself. We turn all the knobs from an engineering and biochemistry know-how point of view, such that the things we are interested in are capturable and differentially detectable, and everything else looks relatively either different enough, or because of the polarity issue, doesn't even go near the pore.
Well, what if you wanted to, I don't know, study a substance that had 2 nonpolar molecules, let's say a carbon dioxide and something else, and you wanted to look at each one individually? Could you do it, or you can only look at polar molecules that'll be influenced by a current?
Right. So if a molecule doesn't have charge and therefore wouldn't necessarily go through the pore as it enters the electric field, we can add reagents that would specifically bind to molecules of interest, adding that charge to them, thereby now allow them to be driven through the pore. Maybe I'll back up one step and clarify that the sweet spot for our technology isn't detecting gases, as you suggest, with carbon dioxide or oxygen, but larger molecules, whether it be analytes, metabolites, drug molecules, proteins, peptides, or even more commonly, DNA or RNA sequences. So being able to identify pathogens, viral or bacterial, for example, based on their nucleic acid content, based on their DNA or RNA specific sequence. That's really the sweet spot of our technology, as opposed to blood gases, for example.
So it's in liquid form that you'd be able to suck stuff into the nanopores, right?
Correct.
It is. And if it isn't in a liquid form in its natural state, we can put it in a liquid form. If we're doing for an ag application, let's say, where we take a sample of a leaf, we can grind it in extraction buffer and put that crude sample into a prep cartridge, thereby having a liquid form by the time it hits the nanopore.
So do you have like an array of billions of nanopores, and you look at the attenuation and how it's different across each nanopore to see what each one sucked in, or do you have just 1 or 2?
Yeah, it's usually a 1 or 2 thing. We're not a high density array like ion torrent trying to do sequencing. We're a low density, modest plexing, anywhere from 1 to 20 on a cartridge, and however many nanopores you need to support that. A single nanopore generally can resolve up to 3 or maybe even 5 different targets from a given sample. So you may want to add more than that for a few reasons, right? If you're multiplexing beyond that, or if you want redundancy to enhance the quantitative power of the analytics, for example.
Well, I mean, in biological systems, I would think that it's rare that you have just, again, one type of molecule. So how do you, again, if you only have 1 or 2 pores with a sample, how would you know that you got the particular molecule you're looking for?
Yeah, so I think it's helpful to think of this in terms of hypothesis testing as the framework within which someone would want to use our technology. So that is, you have a test you're performing, you already know what you're looking for, and let's say it's a panel for some infections, right? Some set of infections, infectious diseases, and the chemistry is already established to have a high sensitivity and specificity for that target if it's present in that sample. So now you're just doing either yes, it was there, or no, it wasn't. Or in some use cases, you want to know a relative amount of a target compared to either another target or some endogenous reference molecule. But in all cases, we're tipping the scale such that the nanopore can pick it up if it's there. Does that answer your question?
What happens once a molecule is sucked into the nanopore? Where does it go, and then what?
It goes into the ether. You never see it again.
The nanopore goes hum, and it burps.
Yeah,
Or
exactly.
eats it. No,
Yeah,
where does it go? What happens?
yeah, so typically, right? There's 10 to the 10 to the 10 copies of let's say all molecules in this chamber above the pore, and there's some critical mass of the ones you're looking for, and the nanopore will capture about a molecule every second, or sometimes a few per second. And so once any one of those molecules goes through that nanopore, it's now on the other side of our chip, essentially in an infinite volume, even though it's a few microliters compared to the size of the molecule, it's infinity. And you never see it again, right? It was the polarity of the voltage that pulled it from the one chamber to the other, and now it's actually for field force reasons impossible to go back the other way. So you're just seeing new copies of different molecules, one after the other serially. And after a few minutes, you've seen hundreds to thousands of these molecules, and you have some machine learning or maybe a heuristic algorithm, depending on the use case. We have both. That says, I saw this critical mass of these two targets. Here's how much we think there is. And these other ones didn't show up at all, so they're negatives.
Okay, so I see why you're saying the hourglass example makes a lot of sense. So how can you tell what has gone through the pore? I know but can you correlate the degree of attenuation of the signal with? Does every molecule have its own unique attenuation signal, or how do you know what's going through it?
Correct. Yeah, that's the numbers game that we're playing, right? We're molecules will be binned according to either a model that was established through support vector machine or machine learning that characterizes every event by some number of parameters. Or sometimes there's very simple tests where you're just looking for this particular, tuberculosis target or two targets. And the nature of those targets, they're distinct enough from the nanopore electrical signal sense that it's a very easy, yes/no question against another control, endogenous control or spiked control that's in there. So we're obviously very skilled at creating that kind of table lookup in nanopore event space to say what you've seen against, again, a finite set of hypotheses.
But essentially, you're trying to become another type of not gas chromatograph, but you're becoming another method of assay that's just like a gas chromatograph. Like each molecule, I would think you'd build up a library of the attenuation signatures of each molecule, and over time you'd have this huge library, and it actually would become a standard over time. Oh, this method of analysis, here's its standard attenuation, and we have, all scientists know this and they reference it. And is that where this is going in one way?
No, it's not exactly that, because remember, there's not a one-to-one mapping between target and nanopore event space, except in that confined menu for that particular set of hypotheses. In other words, you might have one test where the model is looking for 3 particular infections, right? And the model now has these 3 sort of buckets for the targets and what their electrical profile is. And part of the formula, as Trevor said before, we engineer molecules that essentially facilitate that binning, that bind to those targets. In a totally separate test, you can actually reuse those engineered molecules that now, really their only difference is they now have an affinity for a different target. But otherwise, they give you the same sort of letter in the alphabet in that binning of nanopore events. So you don't need this, infinitely expanding category search space, because you can reuse the real estate in nanopore event space, right? The hypothesis might only ever get to 20 targets, let's say. And so we can always reuse the spots that aren't being used because nobody's looking for tuberculosis in a cotton test, right? Where you're testing cotton with a particular virus.
But wouldn't that be nice? Wouldn't that be nice if you could do that? Then you don't have to know ahead of time what you should expect. You can just run it, and whatever comes through the nanopore, that signal means this signal means that. And it seems like that would be a much more expanded, better use of it, if it could be done. I mean, I'm just putting it out there.
For sure, yeah. There's I think for at least the way we think about it from a productization and strategy point of view, rather than trying to make the universal, molecule nanopore categorizing tool, which would be interesting, we're trying to solve specific problems where they need a field deployable product that can give high accuracy, fast, and answer these specific quantitative questions, right?
So what's the accuracy that a certain molecule is there or not, or is it the concentrations of certain molecules? Like, where is the superior accuracy from this method come from?
You want to do some talking?
Sure. So, as Bill described before, the molecule is the nanopore is a single molecule counter, molecules passing through it serially. And so that gives you that higher accuracy in that you're actually doing single molecule counting using an electrical output, as opposed to the majority of lab instruments that look at an optical signal, which is generally an aggregate fluorescence signal. And so by being able to do the single molecule counting, you do get that superior accuracy. And that shouldn't be confused with also limits of detection. And so when you talk about how sensitive can the test be, and that really comes back to our ability to design reagents that are very sensitive and specific, designed to have sensitivity and specificity. So even if your target is in your sample at a very low abundance, our reagents are able to bind to it specifically and then increase or amplify the signal such that you're counting those molecules through the pore and giving you that accuracy.
But I also think part of your question was the qualitative versus quantitative, and both are possible with our workflow.
So, like, I just had a blood test done, and I don't know what they would I don't know. One of the factors was like picograms per deciliter, and they only went to a certain limit below which they couldn't see the substance. So your solution, for instance, could have a much lower theoretical limit of being able to see something in blood or something in some other kind of sample. Is that accurate to say?
Yes.
Okay. Good. I don't mean to grill you. I'm just trying to get a good understanding of this.
Yeah. No. It's okay.
Okay. And is your system you have to bind to certain reagents, or can you just let the native molecules go through the nanopore, or do you always have to bind something to them?
Yeah, like you alluded to before, you can let those native molecules go through the pore, but the pore doesn't give you the resolution to differentiate one molecule from the other. Generally, they give you that same signal as they go through. And so by having those molecules that bind with high specificity to your targets, that's what enables you to have those differentiator, so you know it's your target molecule going through the pore and you can detect it.
Okay. All right, gotcha. I know in chemistry there are like enantiomers, molecules that are left or right-handed. Can you differentiate between those, or are there certain kind of molecules that you can differentiate that no other method can differentiate?
Sure. So, yeah, with the case of enantiomers, if there's a binding agent that specifically binds to one form or the other, then absolutely. So we would use that as the selectivity agent that binds specifically to the molecule of interest, such that when it passes through the pore, we would detect, say, the left-handed form as opposed to the right-handed.
Okay. And where do you see that your system really shines? I guess one way is that it can detect levels of substances far lower than other systems. What are the ways in which it's a better assay than what's out there right now?
Yeah, I mean, I think it it really shines in three different ways. One, as we've been alluding to, it's an electrical counter as opposed to optical. And it does so with these nanopore chips that are small and rugged so they can be packaged into microfluidic cassettes. So this really opens the door to have an instrument that can go anywhere. It can work at low and middle income settings, remote settings. So you can still do diagnostic testing, high precision diagnostic testing, far from any type of centralized lab or even a pop-up clinic type environment. The second is the same sensor, the Nanopore itself, detects both protein or other target analytes, as we would call them, drug molecules, metabolites, but also the nucleic acid of an organism. So that's the DNA or the RNA, which gives really just a ton of information about what you're looking at, if it's infectious tuberculosis versus non-infectious, whether it's drug resistant or not. You can get all of that information out of the DNA. And so by having a sensor that can do really those two large classes, it's what we'd call a multimodal or multiomic, where it can do both molecular testing for DNA or RNA, but at the same time look at target proteins or metabolites, as I mentioned. So that's a second point. And then again, the accuracy that we can get with single molecule counting really is superior to other quantitative methods that are out there. qPCR is a very typical method to do infectious disease viral load, for example, HIV viral load. It's one that's only available in a centralized lab, and also one that isn't as accurate as a single molecule counting approach like the Nanopore gives you.
Okay. So why is it easy for you to create these nanopores? Like, can you create a gigantic array of them, or is it?
Yeah.
I mean, if you're counting like 1 molecule a second, why not have an array of 10,000 of these things that goes in 2 seconds and counts everything in the sample?
Yeah, I mean, that's like I said, interesting thought exercise is the expandability of this and what's possible. But to come back to your first question, the chips we make at wafer scale, and we're working with partners in the semiconductor space to make that a high volume capable process. And it really is a very simple, when it's well engineered, it's a very simple fabrication requirement process and can be very cost effective to make many chips per wafer. And let's say 10 chips goes into a consumable for a particular multiplexed menu. As we look at the price, as you ramp up generating these consumables, the chips themselves will be a fraction of the COGS and the total cost, and so it's a very scalable method, which is required to really have a clear business case to support a given market that you're going to go after. And frankly, that's kind of what's missing in a lot of the thought exercises and research that go on in research institutions, right? Universities, where Trevor and I both have spent a lot of time on that side of it. And you might have the greatest, fastest, most accurate thing out there, but if you can't make it beyond the scale that a few labs can utilize it, then it's probably not going to matter. So it has to meet this scalability requirement, and so that really is why I think a lot of people who came to the company early, and why recruiting for us has always been it's compelling, right? Because there's the story that we've talked about so far on this podcast of what you could do with the thing, and then it's, well, what does it look like to actually make the thing at a scale where you can make an impact? And the simplicity of the sensor and the fact that it works in a scalable fashion is it's critical.
Okay. And then I guess I'm asking for a lot by asking for all these things, but what is the range of molecule sizes that can go through the pore? And would it make sense to have a series of pores of different sizes so you suck the molecule in and then send it through a maze that, silos it further by size or by other characteristics?
So as far as size range goes, we have ways of modifying the buffer to make it easier to see smaller things. Larger things are also sometimes interesting for different applications. Generally speaking, DNA, RNA, these are the targets of choice for the nanopore and the assays that we spend most of our time on. And so
maybe I'll mention the nanopore size we generally use is large compared to other researchers using nanopores. Ours are tens of nanometers. And so at that size, you can fit full proteins through, protein complexes, larger structures. So not as big as a whole bacteria or a whole cell, but certainly large enough to fit larger macromolecular complexes through the nanopore very comfortably. So it really is a wide range of sizes, everything from that small molecule that's a few hundreds of Daltons all the way up to megadaltons.
Okay. So what's the timetable for getting this into production and working on larger sample sizes and really getting it out there everywhere?
Yeah. So we with the current few customers we have and the versatility of the product that we're making for them, we will have a field testing, let's call it a beta unit. Let's see. It's funny, we just did this thought exercise. I would say about 2 years from now, somewhere between now and 2 years from now, will be something that's being tested in the field in appreciable volumes.
Okay. Is it necessary to use like microfluidic, channels along with the nanopore, or is this like a system that could operate without them?
No, that is part of the envisioned use. Is within a single consumable, you have what you need for prep, any amplification if it's required through PCR or any other amplification method, and then some number of nanopore chips. And microfluidics does make sense in that context.
Okay, well, very good. So what's the best way for folks to learn more and, maybe see what one looks like and, speak to you guys about collaboration or questions?
Yeah, I think obviously through the website, we've got a sketch of the technology, areas where we've already demonstrated feasibility. And I think in the coming months, there'll be more information on the website regarding our productization strategy and obviously to the extent that it makes sense to share it publicly in a format like that. But I think that's the best place to start, papers and the website. Okay.
Well, very good. Well, guys, I know I questioned you to death, but I appreciate you being on the podcast, and it's a really interesting idea. I don't know if I communicated that. I just asked questions, but I think it's a really great invention you guys have made, and it's going to be super beneficial in the future.
Yeah, thanks.
Yeah, thanks, Rich.
You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. you enjoy the podcast, please listen, subscribe, like, and share it with friends. Thank you.